like to introduce our first speaker, Dr. Jared Flater. He's a postdoctorate researcher in agronomy at Iowa State University. He earned his master's degree in agronomy in agriculture, and biosystems engineering, and his PhD in environmental science at Iowa State. His work is centered mainly on understanding how different farming practices affect soil health, mainly focusing on natural ways of improving soil through the use of organic materials like compost. Dr. Flater will be teaching a soil microbiology class this fall. Anyone wants to take it? So help me welcome Dr. Flater. Thank you.
Hi, thanks everybody. It's really great to see so many people here that are curious about soil biology. As was mentioned, I've been at Iowa State now for the last 9 years, and most of that time has been focused on exploring more about the primarily soil bacterial community. I just wanted to give you a little bit of a background on kind of the specific research I did. So my master's project, before I went to grad school, I worked at the USDA for an organic agriculture researcher named Cindy Cambardella, who unfortunately she's passed, but she's been a really great mentor to me. And when I worked there, I realized I was naive, I thought we knew everything. But I learned that we don't know hardly anything, especially about soil biology. So that motivated me to go back to school, and when I worked at the organic lab, we did some research and we tried to fertilize our corn crop with 3 years of alfalfa before we planted our corn. And sometimes that worked and sometimes that didn't. Sometimes we used compost and sometimes that worked and sometimes that didn't. So I really wanted to look at the soil bacterial community in particular and try to identify the bacteria we needed in the soil that could help us extract the nutrients from those organic amendments. So I did an incubation experiment where I put some soil in the incubator, so we kept it at like 35 degrees C, pretty warm, and I amended it with compost, alfalfa residue to simulate, plowing down that alfalfa green manure, and then a mixture of those two things. And then I incubated it over time and extracted it at different time points and characterized those communities. And what I learned was that it is really great for promoting that biological biomass. What I also learned is that not very many species of bacteria that were only present in the alfalfa or the compost would persist in the soil. And if I thought about that for a little bit, it kind of makes sense, right? The alfalfa residue itself and the compost are much different environments than the soil. So these bacterial species evolved to survive on the surface of alfalfa or in alfalfa or in a compost pile. And the soil is a much different environment. It's not as much nutrients and things like that. So only a couple species persisted over time through that. So that's just a little bit about what got me here. All that to say is I spent a lot of time looking at soil biology and soil bacteria in particular. And what I've learned is the more I learn, the more I know we don't know anything. So I like to say I'm not an expert, I'm experienced. So I'll try to answer any questions you have, but it's such a huge topic that it's going to be really hard to sort of hit every. I know you guys are going to have a lot of cool questions, and I'm really kind of excited to hear what you have to say. Okay, so soil biology. I mentioned the microbes a lot, but it's not just the microbes, it's that whole community of organisms in the soil. It's worms, it's beetles, it's ants, it's fungi, bacteria, viruses, protists, and they're all working together to help promote nutrient cycling, especially when we're using organic amendments. They're not as important when we're using synthetic fertilizers, and that's kind of what's got us to this point, is we've been using a lot of synthetic fertilizers, which don't feed that microbial community the nutrients they need to thrive. But it does feed our crops, so that's why it's been effective. So first, a little analogy to get back to how much we don't know. Currently, NASA's best estimate is that there is about 100 billion stars in the Milky Way galaxy, which is our home galaxy. And through current research, especially following the development of DNA techniques, our best estimate is that there's nearly 1 trillion microbial species on Earth. That's 10 Milky Ways. If one star represents a microbe, it takes 10 Milky Way galaxies to represent all the microbes on Earth. And that just means and we know maybe about 2 to 3% of those. So there's a huge realm of stuff we don't know that we still have to learn. And that means there's a lot of potential developments that could help us have more sustainable farms and better crop health. So if we think about an iceberg, what we know about soil biology is just a tip that you can see above the soil surface or the surface of the ocean, right? But we know that most icebergs, that bulk of that iceberg is below. We can't see that. So that, what's below the surface represents what we still have to learn. So that's really exciting for me because I love to learn new things and I love to sequence DNA. So it just means there's a lot of questions out there. And hopefully that through this talk and today's workshop, we can really motivate people like you that are out there on the ground to maybe prod their extension agents or research institutions to start learning more about this, because we really haven't focused on it as much as we should be. Okay, so I talked about how all the organisms in the soil are working together. No one species or one organism is doing everything. It's this huge network, and it takes them all working together to do all the things that we need to support nutrient cycling and soil health, and all of which will help facilitate plant growth. So in that diagram here, it's kind of hard to see, I guess, but on the bottom there we have our organic residue. That's sort of what would, whether you have crop residue or you're using compost or manure, that's the input, right? That's where the nutrients that we want to get out for our crops to use. And then we have different levels of the soil food web that have to interact together to make those nutrients available for our crops. So we start with the microscopic actomycetes, fungi, and bacteria, and we move up to the second layer of the soil food web where we have some beetles, mites, protozoa, and then the final layer where we have centipedes and millipedes and larger insects, all of which work together to help support soil health and soil biology. So at the end of this, I'm going to try to get some feedback from you all, because the more I understand about your curiosity, the more I can help direct the questions that I answer in my research. So I want you to think about this, and at the end of the talk, I'll ask for some volunteers, hopefully, to share their experiences. But we're hearing soil biology a lot today. So at the end, I'm going to ask, what do you think of when you hear soil biology? What comes to your mind? Hopefully, there's a lot of different answers to that.
Microbial activity.
That's what comes to your mind? Who said that? Nice. Okay, good. That's one thing. Let's try to get 5 people to say something at the end of the talk. So a healthy soil biology has some direct benefits to our plants and our plant health. One of those ways they benefit that is through nutrient benefits. A healthy soil microbiome can help nutrient cycling. One way that we can support plant health is through mycorrhizal associations, and another way is through nitrogen fixation. So a lot of the nutrients we're using when we're using organic amendments are in organic forms. Nitrogen, for example, plants can only use nitrate and ammonium. In compost and organic residues, manure, there's a lot of nitrogen bound in these bigger compounds that plants can't access. So the soil microbial community's real job is to break those compounds down and finally releasing those nitrogen molecules so that plants can use them. And if we don't have those organisms present, there's not going to be any nutrients available to the crops. And this is particularly important for nitrogen, phosphorus, and potassium. So phosphorus, for example, a lot of phosphorus is bound to the soil particles, and there are specialized bacteria and fungi that produce enzymes that can help release and mobilize that phosphorus and make it available for our crops. So if we have a healthy soil biology, there's going to be more phosphorus available, which if there's more phosphorus available, maybe we don't need to apply as much. The same goes for the nitrogen cycle. This looks kind of complex, but I mentioned before that when we put organic residues on the soil, like maybe this could be alfalfa residue, or even just crop litter itself, or animal manures, these are those bigger compounds I was talking about, and there's that nitrogen right there. We have to unlock that nitrogen molecule from that larger compound, and we do that, or we don't do it, but the soil does that through the activity of bacteria and fungi, and then it enters a nitrogen cycle before we have ammonium and nitrate that the plants can then use. So the more we promote the soil health, the better this process and more efficient this process is going to work. So mycorrhizal associations. Mycorrhizae are fungi that form a symbiotic relationship with plant roots. What this really does is it helps extend the surface area of the plants, of the root system, so it can reach a larger soil volume. And what does that do? Well, one thing it can do is help promote disease resistance. So if those mycorrhizal associations are present, it can actually outcompete some pathogenic bacteria. That mycorrhizal fungi is using nutrients that then limits the nutrients that the pathogens or disease-causing organisms would be using. So that larger soil profile and then those enzymes that fungi can produce help release more nutrients. So it has a bigger volume of soil it's accessing and also releasing more nitrogen and phosphorus from the soil. So they can really enhance nutrient uptake. This is also good for increased water absorption. Same idea, we're accessing a larger soil volume and increasing the available water network or water to our crops. So this might help us, last year this year's a little bit different. We've had a lot of rain in Iowa. I don't know if everybody's from Iowa or not, but we've had a lot of rain. But last year we had an extended dry period in the late summer. So this could be really important for helping us sort of survive through those dry periods, becoming more resilient. Maybe nitrogen fixation or legumes and rhizobia and those nodules you have on your soybeans, maybe that's it might be the most experience a lot of you have with soil biology. So nitrogen fixation in legumes, there's this give and take relationship that happens. The main bacteria that does this is rhizobia, and then they live in these nodules of the legume roots. And what they can do is they can fix atmospheric nitrogen. So nitrogen is the most abundant molecule in our atmosphere, but it's not available for plants to use. So these bacteria can convert that into plant available forms, and they do that in exchange for some sugary substances from the plant itself. So the plant takes the energy from the sun, turns it into sugar, feeds those microbes, and then those microbes in turn take nitrogen from the atmosphere and trade it off to the plant. So that's a really cool symbiotic relationship. And we generally in Iowa, we grow corn and soybeans in rotation. We don't have to fertilize the soybeans, obviously, and in some cases it might be able to reduce the nitrogen requirement for the following corn crop. And that's the same idea with alfalfa in organic systems, where you might grow alfalfa for 3 years to try to really build that organic nitrogen in your soils. Okay, so another way that soil biology can support plant health is through pest and disease resistance. I mentioned a little bit about pathogen suppression, but there are other mechanisms called induced systemic resistance, and then there's even natural biocontrol agents, so naturally occurring organisms in the soil that can help suppress pathogens and diseases. So pathogen suppression, this is a picture of what we would call a disease suppressive soil. So on the left, we have some soils that were identified to have disease suppressive properties, which means there's some bacteria present that can produce antifungal agents in this case, which is the main pathogen affecting these radish seedlings. So on the left, we had the disease suppressive soil, and on the right, the soil that doesn't have those disease suppressive properties. And it's pretty obvious that there was better growth on the ones that had the disease suppressive properties. Some of the mechanisms that works through are shown on the right. So you might hear the word rhizosphere. That's the area that's around a soil root that's influenced by the plants itself. So it's the volume of soil that the plant exudates are influencing. That's what we call the rhizosphere. So that's a really biologically rich area. And if there's the right bacteria present, the plant can send signals essentially to those bacteria, and because they're working together, those bacteria then can produce antifungals that suppress the incidence of pathogens. Now there's also bacteria that live inside the root itself, and those can do the same sort of things. Once the plant tells them that they need to make these pathogens or antifungals, it can do that and then lower the incidence of disease, which is better for both the symbiotic bacteria and as well as the plant itself. So induced systemic resistance is a naturally occurring sort of defense mechanism that plants have. And what that is sort of a signaling. It can either be signaling through the vascular system or it can be signaling through volatile compounds. But what happens is the red area is showing where maybe some fungal or bacterial infections are happening on the plant. The blue is showing where maybe some herb or some insects are eating it, and then the purple is showing some beneficial microbes. So when a plant senses those infections on one side or the other, or the root microbiome tells them that there's pathogens present, it can actually stimulate the plant to increase its defense mechanisms, which will then help support the healthy parts of the plants and make them less susceptible to damage. So natural biocontrol agents, we can help reduce the incidence of disease and pests by outcompeting them for nutrients. That's kind of what I mentioned before. If we have some bacteria and fungi living around the crops that can outcompete those pathogenic bacteria. Or in this case, it's a really cool situation where the blue, hopefully everybody can see this, is like one of the coolest pictures I have, I think. That blue is a fungal hyphae, right? So it's part of fungi, and they're growing in the soil, and they make these little loops, little lassos, and they're just waiting. And when these actually feed on nematodes. So when a nematode is moving through the soil and has the fungi detects those chemicals, it actually will constrict the lasso, capturing the nematode, and then eventually getting the nutrients from it as it dies and decomposes. So that, I don't know about you, but that's pretty cool. So there's another question I'm going to circle back to at the end, and we've discussed soil biology and how it can enhance nutrient uptake and maybe suppress some diseases. And I'm asking you to think about your own experiences on your own farms. If you've noticed anything where you've done some sort of soil health promoting practices, if you've noticed firsthand some differences in sort of disease or pest presence. So we know that soil biology is really important for nutrient cycling and nutrient uptake and plant disease resistance. So what are some of the ways that we can actually make changes that help promote soil biology in this diverse soil community? And if you think about it, it's really about food, shelter, and water for the microbes. And ways that we can enhance those three things are through no-till farming, soil cover, whether that's cover crops, or if you're a small vegetable producer, it might be mulch or leaving residue behind, and then adding compost or biologicals. Okay. So one of the main benefits of no-till farming is that it helps keep our soil structure intact. We talked all about these microbes, right? But what they really need are those soil pore spaces to live in. They live in those little pockets of either water or air, and if those aren't present, we have no habitat for our soil. So no-till is a great way to maintain soil structure. So enhance soil structure and water retention, so we keep those pore spaces. A lot of our bacteria also live in a wet environment, so the more water they have present, the better off. And all of this leads to more microbial diversity and activity. The more diverse our microbiome is, the better we are going to be to do all the things that I mentioned before. The next one I want to talk about is cover crops. Cover crops have a similar effect in providing shelter. So they keep the soil sheltered to help retain moisture, but they also do a good job of enhancing microbial diversity by helping build those pore spaces. Those cover crop roots can go deep into the soil, and also the cover crop roots are there when our crop roots aren't. So that means they're producing sugars, feeding those microbial communities a longer period of the year. So that's the food part of giving them food, shelter, and water. Similar to no-till farming, cover crops also can help promote soil structure, and then with that enhanced structure, enhanced water holding capacity, we have a more diverse soil microbiome, all of which will help contribute to nutrient cycling. And this is kind of one of my favorite parts, is the compost and biologicals. So we talked a lot about feeding the microbes, and that's what compost does. It's exactly what the microbes need to thrive, and then they can in turn break that down and make it into plant available nutrients. One of the main ways they do that is through the addition of organic matter, and this isn't particularly important if we're removing a lot of our above ground biomass, adding a big, and we don't have to necessarily add compost every year. There's been a lot of research showing one big application every 5 or 6 years can be just as effective as doing it once in a while, or once every year, or every 2 years. So this compost is generally just chock full of microbes, bacteria, fungi, viruses, protists, all sorts of stuff. Hopefully we'll see some cool stuff on the slides to really show how full of life it is. And that's all it's all a chance to help inoculate our soils with more microbes. So what are the tangible benefits? Hopefully, through all the things I mentioned, improved nutrient cycling, pest and disease resistance, we can get reduced input costs, less fertilizer use, less pesticide use, and finally, improved crop yields. So yeah, I just mentioned all the ways, right? Enhanced nutrient availability, maybe less fertilizers. More disease resistance and suppression, reduced pesticide needs. Improved water retention, maybe a reduced irrigation or a more resilient cropping system through periods of drought. And then, yeah, again, improved yields. So that was some corn that we grew when I was working at the USDA, and those are some heirloom tomatoes I decided to grow one year at my mom's house. So in conclusion, some of the key benefits of a healthy soil biology and healthy soil microbiome are improved nutrient cycling, disease suppression, and better plant health. The practices I mentioned that we can do to promote these are reduce tillage, covering the soil with either cover crops or mulch or residue, and then organic amendments through compost or manure. So I mentioned we have a lot to learn, but one of the cool things about this is the advent of DNA sequencing. So before we could only understand what we could grow on a plate, and maybe only 1% of soil biology will grow on a plate if we try to culture it in the lab. But now we can sequence all of the DNA in the soil and learn what's there. But another thing I want to leave you with is the importance of farmer collaborations, right? So we have a lot of products. Understanding how they work is going to require collaborations between farmers. So if anybody wants to test some of these things out and really know what's in them and how long it persists or stuff like that, please get in contact with me. I'd love to see if we could set up some trials. Okay. Somebody tell me what they think of when they think of soil biology. Any volunteers? We had microbial activity earlier. That was good. Anybody else? Yeah. Okay. The glomalin, yeah, that's really good for enhancing soil structure. And that's something that bacteria produce they need that sort of like cozy warmness to survive. So they make that when they find some nutrients or something like that, they kind of encapsulate it with that gooey stuff. Anybody else? Yeah.
I took a class through Jeffrey Goldfeld's, and so we add mycorrhiza to our transplant before we go out.
Okay.
And he says he also top dress with manure every 2 years to maintain the bacteria movement. So
Sure. Yeah, that totally makes sense. Yeah. Got to give them the food that they want, right? They need that organic matter, which manure and compost are chock full of that. Yeah.
Is anybody doing research to find out which corn varieties and bean varieties are still communicating with the soil?
I'm not familiar with anything exactly on that, but if I can leave my email, I'll have that on the last slide, and I can do some literature review and see if whatever research has been done, if there has been any. I don't feel like off the top of my head they have kind of looked at that exactly, but who knows? It's possible. Anybody else? Okay, now any actual real world examples of, so you mentioned using mycorrhizae when you did transplanting. Did you notice a difference in maybe the survivability of your transplants with that approach?
yeah, we have 10 acres urban. They had 4-foot gullies down to hard clay. And in 7 years, they're at 13.2% recovery.
Okay, that's pretty good.
It contains flash events.
Nice. Okay, that sounds like it's been pretty good. Anybody else have some firsthand experience? You've noticed any reduced diseases where you maybe have manure or cover crops compared to where you don't? No? Okay. Cool. Well, thank you so much for listening to me. Anybody else have other questions? I think we have a few minutes, maybe 1 minute. 5 minutes? Yeah. Yeah.
In the picture you showed with the little cups of soil, and one was like disease resistant and one wasn't. You said that the bacteria producing an antifungal. Do you think that was a specific antifungal, or like could that also be bad for microorganisms?
It's probably a specific antifungal, but I can't say for sure. I'd have to read that further. In that situation, it's probably better to have the antifungal capacity because of that pathogen. But as soon as that goes away, they'll stop producing that antifungal, and then whatever fungi that are present that might have been impacted by that wouldn't be impacted anymore. So it would hopefully just be a temporary response until that pathogen is suppressed or reduced or something like that. Yeah.
We inoculated some of the transplants going out. Another area we did not because it ran out. And we did a lot of composting, and where we did not inoculate mycorrhiza, we had a lot more parasitic fungus.
Okay.
Stress our plants.
Sure, yeah, that totally makes sense. When you have the inoculation and then using a lot of compost, you're really promoting that healthy soil biology. And when you don't have that, it's a chance for those pathogenic ones to exploit that gap, right? So nature doesn't like gaps. And so when you have that gap of soil biology, that's a chance for the pathogens to come in there and just, boom, have a direct impact, what you saw, that's really cool. That's exciting.
It burned the leaves off some of those starter trees.
Oh, really? Wow.
Yeah, it's food for them to keep track of stuff.
Yeah.
Start over, not too old.
Yep, right. Any other questions? Yeah.
Hey, you mentioned all the diversity, obviously, of all the different organisms. I was curious how, you mentioned crop rotations, obviously, with soy and corn. With cover crops, are there certain mixes or things that we should be considering or thinking about as opposed to just rye, just wheat, something like that, to diversify that to also help with diversity of organisms?
Sure, yeah. I would just say one is better than none and more is better than one. So the more you can do, the most that's feasible is probably going to be your best bet. All those different varieties of cover crops are going to produce different compounds, and that's just going to provide different food sources for different species. If we had a potluck with only one dish, we're only going to get a few people interested in coming to that potluck. But if you have a huge smorgasbord, you're probably going to excite all sorts of people to come there. So that would be my analogy for how a diverse cover crop mix could be better than just a single species, especially if you have a legume mixed in there with that. That would be probably really powerful. Yeah.
So, as far as seeing some observations from using compost too, I put it around some sour cherries.
Okay.
In an organic certified area where I do some of my sour cherries. And the plants that I used Johnson Sioux around the base of 1 year ago have held their leaves longer. My sour cherry trees are bushy, they're bush. Right?
Sure.
From Canada. Anyway, they lose their leaves every summer about this time.
Okay.
Because of our community here in Iowa.
Sure.
So the trees where I put the Johnson Sioux around the base of the plant 1 year ago have kept their leaves longer.
Okay. Wow.
Losing their leaves. So that caused me just to wonder a lot of questions. Am I going to see bricks that's different in those leaves? Or
Sure.
why is it that I'm seeing this?
Yeah.
I'm more confused than before, but it's definitely encouraging.
Yeah, no, that sounds really exciting, and I can't speak to why necessarily that's happening. So I'm not a horticulturalist, so I don't really know a lot about fruit production or things like that, or if the leaf drop was from our humidity and maybe fungal presence. We have a lot of oftentimes increased fungal presence when we have really humid conditions, so the airflow is really good around some crops. So maybe the Johnson Sioux helped outcompete those pathogenic fungi and allowed the leaves to retain themselves, or maybe they were dropping leaves because they're dried out. I don't know, and maybe the Johnson Sioux helped retain some soil moisture that helped them hold on to their leaves longer, or maybe access to more micronutrients in the soil through the fungi that's present in that compost. All of that most likely is working together to make the plant healthier and happier. So that would be my best guess as to what the mechanism is. But I would love to take some soil samples and, and do some analysis of that. It sounds really cool. Any other questions or comments? Yeah.
In the nutrient reductions suggestion that was put out, 10 or 12 years ago, we've not made any progress in slowing down what I guess we refer to as a leaky system here in Iowa that all
Sure.
our nutrients are escaping.
Yeah.
And there's, in an acre of ground, there's probably 12,000 pounds of resident nitrogen there, and then we add
A lot.
120
Or more, yeah.
But does the population of these critters in that first foot of soil keep that nitrogen balance in some sort of equilibrium to keep it there?
Yeah.
And if you're short, does it then leak out and get into the water?
It certainly could. So generally, soil microbes are carbon or nitrogen limited. If you have enough carbon present in the soil, whether that's through crop residue or cover crops, that extra nitrogen probably is going to be immobilized. They're probably going to use that, take that nitrogen up into their bodies, the microbial biomass, to then try to break down the carbon that's present. If there's not enough carbon, they don't really have a need for that nitrogen. So they just let it go. There's no bacteria there present doing any jobs that need that carbon. So if you have the carbon there, then you have a chance to immobilize that excess nitrogen. Does that answer your question a little bit? Cool. All right. Well, thank you, everybody. I appreciate your attention. It's been really fun talking to you.